Hello ladies and gentlemen, I am Dylan from C.A.R. Entertainment and welcome back to more biology revision videos. This time we're going to go over nucleic acid and AS1 biology, tailored towards the CS spec of course, and let's waste no time and get straight into this. So we're going to start to talk about the structure and the function of nucleic acids. Firstly, I want to show you, move your attention to the diagram on the right. Hopefully you can see that fair enough. You can see a circle with P in the middle. That is your phosphate molecule. You'll see your base, which is a nitrogenous base, and then you see your deoxyribose sugar. Now on towards your left of the diagram, that is just another way of putting that of what I just said. But let's go into the information on your screen right here. Basically, to make a nucleic acid, you have to have subunits, and a subunit of a nucleic acid is called a nucleotide. And a nucleotide basically consists of a pentose sugar, or your deoxyribose sugar, as we see here, your phosphate group, and then your nitrogenous base. These three components are combined in a condensation reaction to form the nucleotide. There will be a phosphodiester bond forming between the sugar and the phosphate of the next nucleotide. That is very important. So it's the sugar and the phosphate of the next nucleotide. So if you look at the bottom left point in the deoxyribose sugar on the diagram to the left, there will be a bond coming down from that into another circle, which will be a phosphate, and that bond will be a phosphodiester bond. Then you have, and then basically nucleic acids, where multiple of these nucleotides are together, will be broken down by hydrolysis to then form nucleotides again. Now we're going to talk about different types of nucleic acids, and one of them is DNA. You'll probably be very, very familiar with this. Basically, DNA is two anti-parallel strands held together by hydrogen bonds between a base and bases, between adjacent bases. And if you don't already know, DNA stands for deoxyribose nucleic acid. They also have pairing rules, so adenine only pairs with thymine and cytosine only pairs with guanine. If you want me to break that down, it's A pairs with T and C pairs with G. And then also DNA is a double helix, but you don't really know about the Watson and Crick model, etc, etc, etc on that there. All you need to know is it's a double helix. Now we're going to move on to RNA, one you may be less familiar with, and basically RNA has a sugar ribose, and these are single-stranded instead of double-stranded. RNA is invariably much shorter than DNA, and they have a base uracil replacing the base thymine. There's three different types: messenger RNA, which will carry the code from the DNA in the nucleus to the ribosomes in the cytoplasm, and this is where the protein synthesis will take place in these ribosomes. You also have transfer RNA, which then carries the amino acids in the messenger RNA, where the protein synthesis will take place, as we know that in the ribosomes, and it takes to a single folded in a clover leaf shape. You also then finally have ribosomal RNA, which is made in the nucleolus, and this forms over half of the mass of each ribosome. Here's a wee table. So for an example, the subunits of DNA RNA, you have deoxyribose and thymine, ribosome and uracil. You have the length, very long, relatively short, strand, double strand, single strand, and then base pairings you can see there also. Now we're going to do DNA replication. So basically you'll start off by talking about the DNA is a stable molecule, and replication is the process which replicates DNA RNA. So basically the enzyme DNA helicase will unzip two strands of DNA, breaking the hydrogen bonds. These original strands that have been broken will now become templates for the formation of two DNA molecules. Three nucleotides will link to the template in the correct sequence. So these three nucleotides will come in and attach to the original strands that have been separated. Then a new strand will be joined together by the enzyme DNA polymerase, and this will allow each new DNA molecule to contain one original and one new DNA strand. This is known as semi-conservative replication. Basically you'll have the original strand that has been separated by DNA helicase, then new nucleotides will attach to that there, forming a new DNA strand, and together they will be joined together by DNA polymerase, and then you'll have two sets of these, known as semi-conservative replication. Finally, we're going to talk about Meselson-Stahl, and they devised an experiment in 1958 which grew bacteria in a medium in which nitrogen was supplied in the heavy non-radioactive isotope N15. N is for nitrogen. DNA in bacteria became heavy in generation 0, and then it was transferred to the normal isotope nitrogen-14. But before changing the medium, and then at regular intervals when they had done, they removed samples and extracted the DNA. Then they extracted the DNA, and this involved a density gradient centrifugation, where heavier molecules were located at the lower level in the centrifugation tube. At generation 1, there was an intermediate level with 50% heavy and 50% light DNA, but then after 2 generations, you had 2 bands of 50% intermediate and 50% light. Hopefully this has made nucleic acids a lot more understandable for you. I've been Done from C.A.R. Entertainment, and I will see you next time.